that we can go and get coffee. So, well, my title, my talk, sorry, it's going to be Disentangling Microbiome Effects on Plant Development and Life History. So, when I started this project like 3 years ago at UC Irvine, we were interested in trying to understand how microbes can affect flowering time in plants. And this is a really crazy and amazing question because you wonder like why plants will pay attention to microbes to develop or to trigger ontogenetic transitions, right? So we wanted to understand whether this process was adaptive to the plants to pay attention to the microbes or whether it was adaptive to the microbes to make a plant to flower or to germinate or whatever. But then we soon after realized that it was way more complex, that microbiomes affect way other, more other traits than just flowering, and that they can be actually drivers of the whole plant life history. So if we think about plants, we know that they are not alone. We know that their phenotypes will be the result of the genes, of course, but also the interactions with the environment and with other organisms, including microbes. So after several years of research from groups all over the world, we know that microbes can affect the growth of the plants, and in turn, plants would affect the assembly at the community level in the rhizosphere or in other parts of the plant. But at the time of this, when we started this project, we didn't know, for example, if the evolutionary history of the plants can be reflected in the composition of the microbiomes, and also when these microbes will be affecting fitness or life history traits, those shaping plant performance. So at the end, my postdoctoral time, instead of focusing only on flowering time, it moves around this change of 3 projects. So first, trying to understand how plant evolutionary history shapes microbiome assembly, then trying to understand how microbes affect development and fitness and quantify those effects, to finally try to see if we can evolve, if we can optimize those microbes to perform a particular function. So I'm going to start with the first project, and this one started with a really simple question. That was, are microbiomes really shaped by evolutionary history? And we expected the answer to be yes, but we didn't know how predictable that change was. So for that, we use the model of plant domestication, which is an amazing model. And some of you, or many of you, will consume these vegetables on a daily basis. And this is one of my favorite examples because these all vegetables come from the same plant. So this is the Brassica oleracea plant. And when we select this plant based on the terminal buds, we get cabbages. When we select based on the leaves, we get kale. And when we select flower clusters, for example, we get the cauliflower. So you can see how extreme these phenotypic effects are. And this is really, besides the biology of this is a really beautiful example of how biology and human populations can interact with each other, right? So this system is super powerful because it is recent, it is repeated, and it is well documented. And besides, we can still find living in the same place wild populations of the plants and domesticated versions of that plant. So it's a really good system to ask questions regarding microbiomes or other type of questions. So in terms of the microbiome, when we started this paper, there was this highly accepted hypothesis that every time that you compare a wild plant against a domesticated one, you will always find higher diversity in the microbiomes in the wild plants. And this hypothesis, at some point, it made sense to me, because if you think on the domestication, you know that there is like a genetic bottleneck, right? Every time that you select the plant, you lose some genetic diversity. So people thought for many years that this is going to be reflected in the microbiomes. So we did a meta-analysis that was published last year. So we looked for all the information available on the literature on experiments when they grew wild plants and domesticated ones under the same environmental conditions, and they inoculated the plants with the same microbes. So the expectation was to find differences, but to find higher diversity in the wild plants. We did find that for most of the cases there is a difference in terms of diversity, but for most of those cases that were different, actually the domesticated plants were the ones that harbored the most diversity in the microbes. We are still working now on how that reflects on the functional level. So we can say from this part that there is no simple universal loss of diversity in consequence of domestication, and that the effects seem to be more crop specific. So yeah, so we can say that domestication is a powerful experiment in plant microbiome evolution, and that plant evolutionary seems to shape the microbiome assembly, but not in a predictable way. So then we knew that this different plant evolutionary history would reflect on different microbiome assemblies. So now we wanted to understand how those differences matter or not for plant life history, and how can we quantify those effects. So this is the question that we try to answer in this part of the project. Like, it's again a simple question: how do microbes affect plant growth, their development, fitness, and life history? And this is how life history of a plant looks like in a really simple way. So we have different stages that will be conditional on the previous step, meaning that for a plant to flower, it needed to grow and it needed to germinate at first, right? So from several independent studies, at that time we knew that microbes can have an effect on germination rates, can have an effect on growth via nutrient acquisition, solubilization, etc. And they can also impact the number of fruits in some crops. But we were wondering if there is like a way in which we can include all, like the whole life history trajectory, to try to analyze how these microbes are affecting the plants. So we come up with a combination of two different methods. The first one, it's like a classic model that is used in life history in plants, that are the Aster models. These are really old models that I don't know why people just stopped using them. I think they're pretty cool. And what they do is they try to estimate fitness from all these connected life history stages. And then, together with our collaborator in Princeton University, Jessica Metcalf, she's an epidemiologist, and she came up with this demographic model that tried to estimate how timing in reproduction will shape the optimal flowering time. So to test, to fit these two models that we come up to do like this demographic framework, we did this pretty simple experiment. So we went to the coast of California, and we sampled microbes associated to this plant, that is the black mustard. It's an invasive plant that is phylogenetically related to Arabidopsis thaliana, that is our plant model. So we sampled microbes, we inoculated the plants, and we let them grow. We let them grow under controlled environmental conditions, and that allows us to quantify all these ontogenetic stages. We were able to quantify germination, bolting, flowering, number of flowers, etc. So with that information, we fit our models. And so in here, I'm not going to get into much detail, but here you can see the results of the Aster models. And what we can see is that different microbial communities will have different impacts on fitness. Some of them will promote a higher fitness, while some of them will promote a really, really low fitness. And the effects that we see are not only in terms of this. We can also see how microbes change the size based on the rosette on the width of the rosette of the plant. They also change the survival rates, and they also move the optimum reproduction timing. So this is a contribution that we just submitted last month, and that we are really happy about it, because we think that this framework will allow other people to quantify the microbial effects on the entire life history path. And just to say something about the applications of this type of stuff is that in terms of crop improvement, for example, we can identify microbes that shift the traits that are important for agricultural purposes. In terms of conservation also, we can test whether, for example, native microbiomes can improve or not the establishment and reproduction of certain plants. In terms of evolution and ecology, we can ask whether these microbes can alter selection for plant traits, or even predict whether design communities can improve overall fitness. And finally, then by the end of, well, it wasn't by the end, but by the middle of my fellowship, after we knew already that different microbes have different effects on life history and that we can quantify those effects, then we decided to go for the question of, can these microbiomes be selected, can be evolved to promote a particular function that we know can happen in this part? So if you were wondering what was the risky part of my fellowship, this was. So what we did was we took inspiration from microbial ecology classical approaches of community assembly. So in community assembly, is super common to do transfers of microbes using liquid media. But we tried to do that using plants. So the idea here was that we already had some microbial communities that accelerated flowering from the first experiment. So what we wanted to do was to do a directed evolution of whole communities in planta. And so this experiment went like this. We had communities that accelerated flowering, communities that delayed flowering. And so we sampled some plants with that phenotype, and we used those microbes to inoculate a new generation of plants. From that generation, we selected only, in this case, the fastest or the slowest in this case. And we did that for several transfers, like trying to emulate what people do in microbial ecology in community assembly. So it's important to say here that the target of selection in this type of experiments is the plant phenotype. So we are kind of going blind in terms of the microbial communities that we are selecting. So this is what we got after 4 transfers. And this is super complicated because this plot that looks super simple took me a year and a half of experiment. So, okay, so you can see how we start with super fast communities in the early flowering and kind of late communities in the late flowering. These are controls on inoculated plants. But you see how early differences erode really quickly and how the effect is not that predictable. So we were really disappointed on that result, but then we started to think why this is happening. And so this is, you cannot see, okay, it doesn't see, but this is a figure from a paper from Alvaro Sanchez lab. And so this is a fitness landscape that can be applied also to microbial communities. So you can see how different combinations of abundances of a species can have a different community function. And this community function can be whatever you want. In our case, it's flowering time. And so the idea here is that we are with the directed evolution experiment, we are trying to move these communities around this landscape. But ecological attractors that are these stable points will prevent us to move around the full landscape. The other issue is that it's super easy to lose diversity. And for some cases, when you lose diversity, you also lose the function. But selection is not the only approach. We can also create communities from the bottom up. And so starting again with the communities that we know affect plant flowering time, we can get isolates and make combinations of them to promote the same effects that we are looking for. And I'm not gonna go into much detail into this, but this is so a figure in which in this line, it's what we were expecting if the isolates effects were additive. And what we can see is that they are mostly additive, meaning that the function will increase as you add more and more isolates. So we are still working on this. We're working on the metabolomics and the metagenomics to get insights into the functions behind these phenotypic effects that we see. So I'm going to skip this one, and I'm going to go to take-home messages. So we can say that plant evolutionary history leaves signatures in the microbiome, that domestication reshapes microbiomes, but not in a universal way, that microbiomes are capable of change of affect the whole plant life history trajectories, and this effect can be quantified across development. That microbial effects on single traits will not always predict the fitness outcomes, and that's why we think that demographic models are essential for understanding how these microbial effects are evolutionary meaningful or not. And finally, that microbiomes can be functional and potentially evolvable components, but selection at the level of communities seems difficult because of ecological attractor and other things. Finally, I want to thank my lab at UCI with Maria Rebolleda Gomez, my PI, other collaborators in EEB, Jess from Princeton, and I also wanted to thank the HFSP because thanks to their amazing support, I was able to work on my own independent line, and that's why I just started my lab in Mexico a month ago. So thank you.
Thank you, Alejandra. Very good.